about our method, the results, and also our case study. We will discuss about it, and of course, to conclude about it too. So first, I'm beginning with the citizen science. What is it? It's a form of production of scientific knowledge in which actors of civil society participate actively and deliberately. The citizen science has several benefits for the local population. So first, it increases, of course, the involvement of people, their interest also, and the quantity of data for scientists. It also reduces the cost for scientific research. But why I'm talking about citizen science today? Because we will focus on one NGO called NatureDive, which is located in Cannes. Their goal is to observe the marine environment by using a specific method. It's the method FAST, belong to the citizen science. The method FAST is a method of fish observation that I will discuss about it more deeply after. And our topic today is to see if this method FAST could be relevant for the scientific research. So this is here the main questions of today that you have to keep in mind. Is the FAST method a relevant protocol to measure the biodiversity? So let's begin with the introduction. FAST means the fish assemblage sampling technique. It's observations of different species of fishes used by divers. So here you have a list of 23 fishes belong to a high level of trophic chain. They are also vulnerable to fishing activities. So if they are present in the ecosystem, their function is active and the food chain is preserved. So they all have specific function. The protocol consists to do 6 observations during 15 minutes just by looking the presence or the absence of the organisms. If the organisms is present, then a score will be assigned to him.
Okay, so the main goal of the FAST method will be to measure biodiversity in an area. But there is plenty of other ways to measure biodiversity, and one of them is the Shannon index that we are going to show you in this presentation. So first, why have we chosen the Shannon index? It's because it's both sensitive to abundant species and rare species, and that was the case in our set of data. But first, we need to understand what is biodiversity, and I will try to explain you that with this little example. So, in order to understand what is biodiversity, we need 3 different criteria: the abundance, the number of species, and the evenness of the distribution. Abundance is the number of individuals inside the community. So for both community, it's 10. Number of species is, for example, an octopus, sea urchin, fish, and a shark. It will be 4 species in each community. After that, the evenness of distribution of species is more the way that the species will be distributed inside the community. And for example, in community A, you can see that the shark species seems to be really overwhelming over the other species that is not really represented. But in community B, the representation is more equal. So by doing the calculation on the software R, it will take into consideration the 3 criteria and give us a Shannon diversity score. And this score will give us a hint about the biodiversity level. And the more higher is the Shannon index score, the more higher the biodiversity will be. And for example, in our community A, Shannon diversity is only 1, and Shannon diversity is more higher in this community, so we can lead to the conclusion that community B seems to be more biodiverse according to the Shannon index. Okay, so now we can go more into details in our methodology. The first step for us to get the good data was to assess which fish were present in both our data set that we collected on Rance Island and also in the board FAST. And we are left with only 9 species of fishes. So in order to increase our set of data, we have decided to put every species or races that we have seen during our trip on Rance Island in the RAS category. After that, we have created the FAST score by summing every little score that you can see there for one site. So yeah, sum of the score. And after that, we have to do the same for the Shannon index.
So then, we use also a moving average. We need a transformation of our data because the method FAST and our sampling data during the expeditions are different. So as I said earlier, the FAST method is composed of 6 observations per site, and our sampling method is only 1 observation per site. So here you have a map of Lierans Iceland with the red dot represent all the sites during the expedition. So how did we proceed to create this mean? So first, we did the average of 6 sites for only 1 site. And after, we moved the average and we applied the same process on the following sites, like this. And this was done on both Lierans Island, on St. Marguerite and also St. Honora. This moving average is very important and necessary to manipulate our data. And so we applied on both on our FAST score and also on our Shannon index. So then, the results part. So here it's a little reminder about the main question of today. Is the FAST method a relevant protocol to measure the biodiversity? So to reply to this question, here you have a plot with in x-axis the moving average of the Shannon index, and in the y-axis the moving average of the FAST index. So at the view of the plot, the dot seems to follow a linear model. So if you pick a high value of FAST index, we will have a high value, for example here, we will have a high value of the Shannon index. So we decide to check this indicative guess by applying a correlation test. And also to see if there is a relationship between the FAST index and the Shannon index. This test was applied under the software R, where we did all our statistical tests during the project. So this test gives us 2 main information. The first one is the R squared, the coefficient of determinations. It's the percentage of the data variation given by the linear model. So here we have 83% that determines the distribution of the score toplet. So we can say that there is a huge fit between the FAST index and the Shannon index. Then we have a second information, which is the p-value. The p-value gives us the significance of this relationship. In statistics, there is a threshold of the p-value, which is 0.05. If the p-value is under it, then the results are significant. But it's if above 0.05, then the results are not significant. And here, our results are far under 0.05. So we can say that the p-value confirms the positive relationship between the 2 variables. So according to these powerful results, we are going to apply this on our case study.
Okay, so we have seen before that we have a strong link between the FAST index and the Shannon index. But this is a theoretical link, and we need to do it in an actual case study to see if the link will be still present in reality. And so the first observation will be that we know, according to the literature, that the species richness, which is also a biodiversity index that takes the number of species on an area, is greater on St. Honora than in St. Marguerite. So, because there is a strong link between the 2 index, we can make the assumption that they will follow the same trend if we compare the 2 islands. And this is the assumption. So, in order to test it, we have divided in 2 different tests. And the first will be to compare the mean of both islands for the Shannon index. So in the y-axis, we have the mean of the moving average of Shannon index for both islands. And you can see on the graph that it seems that St. Honora is greater in terms of mean than in St. Marguerite. So we can put our assumption that Santana seems to be greater, and we can test it. And to do it, we are using a student test. So by student test, that will compare the mean of two different samples. And for this, we have obtained a p-value that is below the threshold of significance. So we can conclude that biodiversity level is significantly greater in St. Honora than in St. Margaret with the Shannon index. Also, we can think that because there is a strong link between the two methods, then for this time the result for the fast method will follow the same trend. And that leads us to the second test, but this time with the fast method and with the mean of the moving average of fast score for both islands. And this time it seems also that St. Honora is greater than St. Margaret, so same assumption. And however, this time we didn't obtain a p-value that is significant, so we cannot conclude that the biodiversity level is higher, greater in St. Honora than in St. Margaret according to the fast score. So it's quite strange that we have obtained this result, so we are going to try to give some ID for why. So the first is the size of the sample. That could have an issue, because we were able to use only 9 different species of fishes in our data set. It's maybe too little to have significant results. Also, the fast method was originally developed for diver, and in our case, we were not diving, but on a kayak. So we were not able to explore all the water column, but only the surface. And after that, the way that we have done the observation, so only one observation, and we need 6 observations to be relevant with the fast method, is maybe an issue, because we were using a moving average that has transformed a lot of data, and that could include mistakes and some issue with the results. And the last one is maybe the Lance Island that have an issue, and we need, in order to be able to say that, okay, the method is not significant, we need to do it on other islands and test it on other places.
So to conclude, here this is a little summary. So first, the fast method, as we know, assesses the biodiversity. Then we decide to compare it with the Shannon index, and we found a very strong correlation between the Shannon and the fast method. So we decide in our case study to apply this, but unfortunately we found results that are not significant. So as Ludovic said, maybe we need to improve our experience to apply correctly the fast method. So thank you very much for your attention.